All right, so it is 2. We will get started here. So just as a reminder, next week, Friday, is our first test. All the material that we'll be covering on the test is in our test 1 folder. So make sure you go over the questions we go over in class. Make sure you answer those. Good way to study also is those study guides I put up. If you can answer those without looking at your notes, you should have no problem answering anything I can give you on the test. Look at the old tests. Be aware that the tests that are online, the paper tests, might have extra information that we haven't gotten to yet just because of the speed of classes. So if you see a problem where like we have never talked about it, then I wouldn't worry about that. That's most likely going to be on test 2 for you. So with that, continuing with our lecture from Wednesday, we were talking about how to purify proteins, how we can attack different characteristics of a protein to try and separate those proteins from a mix of proteins. And we talked about salting out as one technique. The next technique is called ion exchange chromatography. Now, the idea of ion exchange chromatography is that you have a solid phase and a liquid phase. And in the solid phase, also called the matrix, you have charged particles. You either have positively charged particles or negatively charged particles. So if you have negatively charged particles, that's called cation exchange. So that's negative particles. And if you have positively charged particles, that's anion exchange. And if you remember, cations are positively charged molecules and anions are negatively charged molecules. So when we talk about exchange, we're exchanging the proteins, right? So a cation exchange will bind positive proteins. An anion exchange will bind negative proteins. And the way that this works is that if you add a sample of proteins, a mix of proteins, as shown here in brown, different proteins will stick to the matrix with different affinities. So let's say that we were doing a cation exchange, right? So let's say the picture on the left, a cation exchange. That means that this yellow protein would be the most positive, and this red protein would be the least positive, because if it's cation exchange, the more positive you are, the more positive charge, the more you're going to stick to the beads. And so this is a very efficient way to separate proteins based on charge. And the way to get knock these proteins off is through salt. And so that's shown on the graph, and the y-axis is cut off, but the y-axis is basically volume. And what you do is that you add high salt to your column. And this salt will outcompete the proteins that are stuck. So to get the blue protein off, you would need a lower salt concentration than maybe the yellow protein. But you just flood your system with salt. This salt will bind to the column, knocking your protein off, and then you can collect your protein. So on the bottom right here, these are the types of columns that are commonly used. CM is usually used for cation exchange because it's negatively charged. DEAE is usually used for anion exchange because it's negatively charged. Right. So the first question I have for you is, I have 6 proteins shown right there in the middle with different PIs. And I do have a poll for this to see where we land. Let me get that up. So we're just going to do cation exchange for the poll, but I want you to rank these proteins in order of elution, and elution means to come off the column. So rank these proteins in order they will come off the column based on their PIs. So for a cation exchange, I would like you to rank the proteins shown in the order that they'll come off of the column. So we're going to take a minute or so to do that. Once you have your order, please put it into the poll. If you have any questions, do not hesitate to let me know. In case you're stuck, remember PI is pH when you're neutral. So really the question you have to be asking yourself is, does a high PI mean I'm more positively charged, or does it mean I'm more negatively charged? And to really think about that, remember the pKa side chains, or the pKa values for our side chain amino acids. Maybe that'll help guide your thinking. And we'll give this about 1 more minute here. All right, so let's take a look at this. So the way you relate pi to charge, and that is charge at 7, pH of 7, because that's what we do our experiments in. The lower your pi, the more negative you are. Because remember, pi is the pH which you're neutral at. So if you're neutral at very low pHs, that means you have a lot of negative groups. If you're neutral at very high pHs, like pH of 13, that means you have a lot of positive groups. So the order of evolution for cation exchange would be protein 4. 4 has the lowest pi, so it's the most negative. Then 6, 1, 3, 5, 2. With 2 having the highest pi, so it's the most positive, and it's going to stick to the cation exchange the strongest. Anion exchange is the exact opposite. Positive proteins will come off first. Negative proteins will stick on the strongest. All right. So any questions about ion exchange at all? All right, let's move on then. Another way to purify proteins from a mixture of proteins is to use the hydrophobic nature of a protein. So here, with this hydrophobic column, it's the same basic idea of the ion exchange column, except this time, instead of charged beads, we're using hydrophobic beads, phenyl or octyl groups. And under high salt condition, proteins stick to the column. And then when you lower the salt, only very strong hydrophobic amino acids or proteins stick. So under low salt conditions, as seen here on the right, and this key is if you have a blue sphere, you're hydrophilic. If you have a orange sphere, you're hydrophobic. So under low conditions, low salt conditions, the more hydrophilic you are, the more likely you are to be eluted off of a hydrophobic interaction column. The more hydrophobic you are, the more likely you are to stick. So in that sense, it's the opposite of the ionic exchange column. In the ion exchange column, we use high salt to knock proteins off. Here, we use low salt to knock proteins off. So same type of question I have for you. I give you 3 proteins. I want you to rank them in order of elution again, if we're using a hydrophobic interaction column. So this will be a good test for you to see how you've been learning your amino acids. So you know your hydrophobic amino acids from your hydrophilic amino acids yet? Or do you know your hydrophobic amino acids from your charged amino acids? So take a shot at looking at these one-letter codes and see if you can tell which amino acids are hydrophobic. So I'll give everybody a couple minutes to test themselves on that. And we'll do like another minute or so for this. All right, so let's talk about some amino acid generalities. So what I want you to be able to do eventually is be able to look at a one-letter code and tell me its property at a pH of 7. So our positive amino acids are K, sometimes H. I'm going to put H there for right now because it is charged a certain percentage of the time at 7, positively charged. Negative amino acids are D, E. Polar, our polar amino acids are S, T, N, Q, sometimes C. Cysteine is somewhat polar, somewhat non-polar. Depending on what experiment you look at, cysteine behaves either way. Non-polar. That's A, I, L, M, F, W, Y, V, and somewhat P, somewhat G. Again, those are kind of odd cases. P, G, and C don't neatly fit into either category because, like I said, different experiments will tell you different results for those. But in general, that's our ordering of amino acids. And if you find something on the internet that disagrees with me, I wouldn't be too surprised because there's still active debate on what amino acid is what. So what we'll elute first, it's the thing that is the least hydrophobic. So if we look at protein 2, you have a hydrophilic, charge, hydrophilic, charge, hydrophilic. So not really many hydrophobic groups. So this will be first. And then protein number 1. Well, actually, sorry. Yeah, protein number 1. R, so charged, hydrophilic, charged, versus number 3, charged, and that's about it. So it goes 1, there, 2, 1, 3. And all I'm doing is I'm asking myself, how many nonpolar groups are there? The more nonpolar or hydrophobic groups, by the way, those words are interchangeable. Polar, hydrophilic. And hydrophilic is also charged. So I'm just counting how many hydrophobic groups are there. The more there are, the more likely you're going to stick onto our hydrophobic column. All right. So any questions about this technique or our groupings of the amino acids? All right, let's move on. Another way to separate proteins, like I said, separating proteins is all trial and error. So you need to have a lot of different tools at your disposal depending on what protein you're working with. So the next experiment is called gel filtration. And the way gel filtration works is that you have a column that's full of beads. And this yellow thing in the middle is our bead. These beads are full of little tunnels. And you can buy beads with different tunnel sizes. But the idea here is any small protein beyond a certain or under a certain weight can go into the tunnels, while a big protein cannot. And so the way that I always think about this is like, let's say that I have a system of tunnels, right? And sure, goes like that. And I put 2 people in this tunnel, represented by, like, I put a person and I put a giant in the tunnel. I don't know. And at the end of the tunnel, I have a bunch of money. And it's pitch black in that tunnel. Now, the small person can go into these side rooms. The big person can't. And I will say whoever reaches that money first gets it. Well, the big person only has one path to go. They can only go down the hall right to the money, while the small person could go in these little holes, right, and waste time doing that. So the big person is going to get the money first. It's the same idea with gel filtration. Big proteins cannot go through the tunnels and the beads, so they will elute first. They go right through the column. While small proteins, they go through the tunnels and the beads. Therefore, they actually take a longer path to go all the way through the column. So they take longer to elute. So to make sure we have this idea down, I have another short little poll for you. I have 4 proteins, and I give their sizes in kilodaltons. Please order these in the order they'll elute on a gel filtration column. So take a minute or so to do that. And if you have questions, let me know. Well, I guess I don't have to wait too long for this one. Seems like everybody got this one. And it is the easiest concept compared to the other ones, because you don't have to look for like what type of proteins are in there. You don't have to think about what's a cation, what's an anion, how's that really relate to PI. You're just looking at size. So the biggest thing comes out first, and then they just go in order. Yep. So gel filtration column, pretty straightforward. Now let's talk about the best column you can use. It's called affinity chromatography. And the idea of affinity chromatography is that the beads in your matrix are made in such a way that they will only bind your protein. So we're not so much going for different characteristics of proteins. We're just saying we're going to bind our target protein, and anything that's not our target protein goes through the column. Now, how do we do that? Well, there's 2 types of main ways to do affinity chromatography. One is called immunoaffinity. In immunoaffinity, you use antibodies that are attached to the beads. And I mentioned this when we talked about the ELISA, but an antibody is a protein that is so specific, it only binds one target. So if you have an antibody that is made against the protein you are trying to separate, well, you just make a bead with those antibodies and you separate only your protein. Everything else washes through. The other way to do affinity chromatography that's popular is by using metals, metal chelate affinity chromatography. The idea of this is you have a column full of divalent metals. Zinc is usually used, but nickel's also a very popular choice. So you just put a column full of zinc or nickel. Then any proteins that can interact with these metals or chelate them will be stuck to the column. Now, interestingly, this only happens to proteins that have like 5 or 6 histidines in a row. And they're usually at the end or C terminus. And this is called a his tag. Now, that's very convenient for us because in nature, proteins just don't naturally have a his tag. Proteins naturally just don't have 5 or 6 histidines at the beginning or end of their sequence. So we can make proteins that have histidine tags to them using techniques we already talked about, right? You take DNA of your protein and using PCR, right, you can modify your protein to have like 5 or 6 histidines at the beginning or end. So you do that to modify your DNA sequence. You put this modified DNA sequence in a vector and you are ready to go. You infect your cells with this vector, you grow up the cells, you now have a protein with a his tag. You bust open the cells, run all the proteins through a column that has nickel in it. Your protein will stick, everything else washes through. Then you can remove it with salt and you have your protein purified. With immunoaffinity chromatography, usually salt, you can't do that because the antibody binds too strong, that salt won't kick it off. So usually you have to cut off the antibody. So your antibody's on a bead, usually you just snip off the antibody, and then once they're sniffed off, you can separate them through like centrifugation or something like that. But yeah, that's affinity chromatography. It's the best if you can use it. Not all proteins can tolerate a his tag. Some proteins will not work if you put a his tag on them. And affinity chromatography itself is expensive. Buying antibodies isn't cheap. Putting them on beads isn't cheap. So that's why, affinity chromatography might not be available to you. But if it is, I would say you would want to use it if possible. So any questions about the theory of affinity chromatography? All right. Now, once you have your proteins separated, again, proteins aren't colored, so you usually just have a clear tube. And one of the ways to check if you have your protein and to also check that your sample is pure is by running what's called an SDSPAGE gel. SDS is a detergent, sodium dodecosulfate, and PAGE stands for polyacrylamide gel electrophoresis. And the idea here is you take your sample and add SDS. SDS binds to all proteins at a constant rate. I think it's every 1.6 amino acids, one SDS molecule will bind. And since this is a detergent, what it will do is it will unfold your protein. So it'll take your folded protein and make it more or less linear. And it will give it a uniform negative charge because sodium dodecyl sulfate, the head group, is negative. So what you're doing is that you're denaturing your protein. So this is like after you run SDSPAGE, you can't get your protein back. So you're usually using this for just checking if your protein's there. There is non-denaturing technique. And that's just called PAGE. So if you don't add the detergent, you could get your protein back, but it's a little more complicated to run. SDSPAGE is much more straightforward. Now, in addition to SDS, you also want to add 2-mercaptyl ethanol or beta mercaptyl ethanol to break disulfide bonds. Now, the amino acid cysteine, the side chain of a cysteine ends in SH. And what can happen is that if 2 cysteine side chains come into contact with each other, they can form a bond between their side chain sulfurs. And that's called a disulfide bond. And that can prevent your protein from becoming linear. Because let's say, let me get a different color. If you didn't add beta mercaptyl ethanol, your protein could look like that, where you have disulfide bonds holding some of it together. And that would mess up your page. It would run differently than if it was totally linear. So by adding beta mercaptyl ethanol, you break those disulfide bonds. And acrylamide, the way to think about acrylamide is it's just like a net with random holes in it. And then what you do is that you apply an electric current. Since the proteins are overall negatively charged, they will move towards the bottom of the gel. And how far they move is based on how big they are, because they are moving through these random holes. The smaller they are, the further they can travel. The bigger they are, the less they travel. So this is the opposite of gel filtration. In the gel filtration column, big things came out first, small things came out last. In SDSPAGE, small things go to the bottom, big things stay at the top. And by doing SDSPAGE and by running a ladder, and what a ladder is, it's just a sample with proteins of known molecular weight. So you put in a sample and there's 10 proteins in there, and you know the weight of all those 10 proteins. By making a graph like this, which has molecular weight on the y-axis, and then on the x-axis you have distance traveled in the gel, and you make a fit line. Depending on how far your protein travels, you can determine the size. So that's one of the ways that you can test that it's your protein if you know its size. And it's a way to test for purity, because as shown in this lane, we only have one band, which shows that we don't have any contaminant proteins. Compared to this lane, where you have a lot of bands, and that could be a contamination, or this lane. All right, so any questions about the theory of SDSPAGE? And I kind of already gave the answer to this question. Rank how far they will travel down from least farthest and most farthest. So protein 1, most farthest. Then second, third, least farthest. And it's just strictly based on size. But any question about SDSPAGE itself? All right. And you can make SDSPAGE a little more complicated by running what's called a 2D gel electrophoresis. In 2D, the difference is that in one direction, and here I have left from right, you have what we're calling isoelectric focus. So basically how this works, you have different columns in your gel that are different pHs. So this, like on the far left, it could be pH 1, then 2, 3, all the way to 14. And you can run an electric current going from left to right for your proteins. And remember, we said PI is the pH where you're neutral. So in isoelectric focusing, your protein will travel to the pH where it becomes neutral. So it will travel to its PI. Then you can separate by size up, down using our SDSPAGE. And you get something that just has a ton of spots like this. So when would we ever want to use 2D gel electrophoresis? So let's say you are studying a cell and you want to know under different conditions what proteins are created. You want to know all the proteins in a cell under different conditions. So what you can do is that you could harvest those cells and do 2G electrophoresis on all the proteins. Then for each spot, you can cut off that spot and you can sequence it using techniques that have been mentioned in this class. I think we talked about those. If not, we're going to talk about those like next lecture. But yeah, you just sequence all the spots and you can figure out, okay, this spot I have protein A. Maybe under different conditions, I didn't see that spot, which tells me that when I subject the cell to these conditions, I see this protein. So what I want people to do is I want you to take a couple minutes. I want you to draw a square where the left corner is pH 1, the right corner is pH 14. I said in the bottom, but it doesn't matter. And I want you to mark all the proteins on that square, and there's one that is cut off. Protein 5 is 32 kilodaltons, PI of 8. So SDSPAGE page is going from top to bottom. Isoelectric focusing is going left and right. So using that idea, see if you can place where these 5 proteins would end up using 2D gel electrophoresis. So I'll give everybody a couple minutes to do that. All right. So the way I would handle this is I would start with my smallest proteins. So if you go by weight, the smallest proteins are 1 and 5. So they're going to be at the bottom. And protein 1 has a pH of 5.3. So they are neutral at a pH of 5.3, sorry. So 1's like right there. 5 has a pI of 8, so it's going to be more to the right. But they should be on the same row because they weigh the same. Next heaviest, protein 4 at 55, same pI as protein 1, so same column, but a little farther up. Protein 3 is the next heaviest, and it has the highest pI, so it's going to be the most to the right. Then protein 2 is the heaviest, so it'll be at the top. Same pI as in protein 5, so same column. So generally, that is how the proteins would line up if you ran them through 2D gel electrophoresis. So any questions about that? All right. So it is 2:46. And rather just start a new PowerPoint because I believe this is the last question here. Yep. So we will call it good here. I know I didn't get to the PowerPoint that I had scheduled for today. We're going to do that Monday. And the PowerPoint I had scheduled for Monday, we'll do that Wednesday, which is what I actually thought would happen. So that works out. Just as a reminder, though, we have our test next week, Friday. So make sure you're studying for that. If you have any questions about that, please let me know. Otherwise, I will put up a homework for today. And make sure you do that before the test because the material on there will be on the test. And I will see everybody hopefully next week. Stay warm out there. It's going to be chilly, but I believe, yeah. Anyways, have a good weekend, everybody. So the test info is up to Wednesday's class. No, the test info would be up to Monday's class because the last lecture that's on the test is the lecture I prepared for today, which we're going to go over Monday. So yeah, Wednesday's class won't be on the test at all, but Monday's will be. Yeah, if you're ever confused, just go into the test one folder and it will show you all the PowerPoints on there. All right. Goodbye for real this time.